Hello everyone, this is my research on fermentation, a broad history and nutrition review. My name is Jaclyn Walsh and my mentor is Professor Tammy Basil. What comes to mind when you think of fermented foods? Usually things like sauerkraut, vinegar, and beer are what typically come to mind. In reality, many foods that are considered standard Western cuisine are fermented. Soy sauce, chocolate, bread, yogurt, and cheese are all fermented foods common in Western diets, but we do not usually think of these foods as a function of bacterial presence. These are all live cultured foods, meaning foods that have been fermented over time. And with recent health trends and global lockdowns, people have been keeping themselves busy by relearning ancient traditions of bread making, kombucha brewing, and experimenting with cuisines from different cultures. I am one of those people, and in this presentation, I hope to share my passion with you. So let's examine the historical and scientific context of the process of fermentation, specifically its role in human nutrition. Essentially, fermentation is the controlled decay of certain foods through the application of sugar, microorganisms, and time. This metabolic process results in foods with higher probiotic counts, increased nutritional value, and longer shelf life. In the ancient world, fermentation was widely used to preserve foods through harsh seasons as well as a way to create alcohol. For example, in Egypt, 3500 to 300 BC, where cultures were becoming increasingly agricultural, the production of mead was a fermentation-driven process that used bacterial growth to people's advantage by creating beer and bread. As you can see in the picture here of ancient Egyptians harvesting grapes and fermenting them in clay pots. These staple foods made up the majority of Egyptian diets, and for good reason. In a time where clean water and good harvests were not guaranteed, people needed a reliable method for preserving food safely. In Matt Badcock's Fermented Fruits and Vegetables, A Global Perspective, the author explains fermentation is a relatively efficient, low-energy preservation process which increases the shelf life and decreases the need for refrigeration or other forms of preservation technology. It is therefore a highly appropriate technique for use in developing countries and remote areas where access to sophisticated equipment is limited. The process of fermentation killed harmful bacterial growth and kept foods suitable for eating for extended periods of times, often for years. To ferment foods was so simple and attainable that everyone from peasants to royalty enjoyed live cultured foods. Egypt was not the only place where fermentation was used. Parts of Asia and the Middle East, in 7000 BC and 2000 BC respectively, pickled vegetables and made alcohol from the sugar-laden fruits that grew natively. The reason fermentation has been a part of so many cultures spread across the world is because its natural occurrence in nature, which could easily be replicated by early humans, and the reason it stuck was because of its long list of benefits. Humankind seemed to master fermentation before they even knew how it worked, strictly through trial and error. There was not a true understanding of how fermentation worked until the 19th century, when the work done by Louis Pasteur, along with the invention of the first microscope, led to the development of germ theory. Louis Pasteur was a French scientist who proved the germ theory of disease, gave us pasteurization and the first vaccine, and because of this he is known as the father of microbiology. He came to these discoveries by using the first microscopes and experimenting with fermentation, enabling him to identify the first microbes, recognize fermentation as living yeast, transforming glucose into ethanol, and defining fermentation as a vital process of respiration without air. Not much later, in 1877, Moritz Traube, a German chemist, was able to isolate enzymes within fermentation. And 20 years later, another German scientist, Edward Buchner, discovered how to use lifeless chemicals rather than living yeast to obtain fermentation. Through their work on the scientific process of fermentation, Pasteur, Traube, and Buchner, and many others, paved the way for an entirely new scientific field of biochemistry, all through experiments with fermentation. It is now understood that there are three main types of fermentation: lactic acid, ethanol, and acetic acid. The first two types of fermentation, lactic acid and ethanol, are the most common because they are used for preserving foods and creating alcohols. The third type, acetic acid fermentation, is used to make vinegars, condiments, and kombucha. All three of these metabolic processes start out relatively the same, in an anaerobic environment, one without oxygen, where beneficial microbes like yeast, bacteria, and mold break down carbohydrates and sugars into pyruvate. The next step in the process is where the first two types of fermentation are set apart. If the pyruvate molecules are transformed into lactate, then it is a lactate or lactic acid fermentation, providing us with foodstuffs like sauerkraut, yogurt, soy sauce, sourdough bread, chocolate, cheese, etc. However, if the pyruvate molecules are transformed into acetaldehyde, carbon dioxide gets released and the acetaldehyde gets transformed into alcohol. Although ethanol fermentations end here, acetic acid fermentations require an additional step involving oxidation. In the second step, ethanol alcohol is further oxidized to form acetic acid using Acetobacter bacteria. It should be noted that when oxygen is introduced to a system, in this case a microbial oxidation of alcohol to acid, this is an aerobic process. In other words, introducing an oxygen-rich atmosphere to an ethanol fermentation results in the formation of acetic acid. Another way to look at fermentation processes is by time. Using a lactic acid fermentation as an example, generally in the first 24 hours after the vegetables have been submerged in water and salt, the microorganisms start to kill off bad bacteria. Within 2 to 3 days, the acids begin, and between the 4th day and 4 weeks, the aging begins. From the very beginning of the process, when the good bacteria are introduced, the food becomes more beneficial in a number of ways nutritionally. Lactic acid fermentation, used to ferment fruits and vegetables, has a large positive impact on human nutrition. Our bodies already use fermentation in the digestive process to break down carbohydrates and fibers into useful products in the colon. By introducing live cultured foods in the human diet, digestion is improved in 3 ways: supplying enzymes to conserve energy, good bacteria to increase intestinal flora, and pre-digested foods to provide readily available vitamins and minerals. Digestion takes a lot of energy, and one way the body can reserve some of its energy is by using enzymes. Fermentation products provide enzymes necessary for digestion. This is important because humans are born with a finite number of enzymes, and they decrease with age. Fermented foods contain the enzymes required to break them down. Formatively, enzymes are proteins that lower the amount of energy needed for a reaction to occur. For example, amylase is a digestive enzyme that helps change starches into sugars, and though it's found in your gut, this enzyme is also present in fermented foods. When live cultured food is consumed, the amylase enzyme helps break down other non-fermented foods in the digestive system and has pre-digested the fermented food, lessening the digestive workload. In addition to pre-digested foods being easier to break down, it gives us a way to eat plants that otherwise would have been a waste to us. The most famous example of this is sourdough bread. Ground wheat and water by themselves have little to no nutritional value. However, combining ground wheat and flour, then letting it ferment, allows this food to not only triple in size, but also unlock the nutrients in wheat that is otherwise undigestible to the human body. Sourdough bread provides minerals, vitamins, and antioxidants which would be otherwise untapped without the help of microbes. This is made possible by the microbes in fermentation which degrade phytates. These phytates bind to minerals in whole grains, making them hard to absorb. After lower levels of phytates are achieved, the body can absorb minerals like potassium, phosphate, magnesium, and zinc. The fermentation of whole grains also increases folate and antioxidants. Additionally, fermented foods help with digestion by providing good bacteria, also known as probiotics, that have been found to impact more than just the gut. Increasing the intestinal flora by introducing probiotics through live cultured foods provides benefits such as preventing diarrhea, improving mental health and heart health, reducing allergies and eczema, help digestive disorders, boost the immune system, and may help prevent obesity. Overall, the probiotic benefits of fermented foods promote a healthier, nutritionally equipped body and brain. Fermented foods were once invaluable to humankind's survival before the invention of refrigeration or the industrialization of food production. Then, ever since the discovery of microbes in the 19th century, they were linked to illness and demonized by Western culture. Slowly, we are rediscovering the benefits of microbes by going back to our roots and brewing kombucha, pickling produce, and baking sourdough bread. Michael Pollan, author of the book Cooked, A Natural History of Transformation, is an avid supporter of reincorporating live culture foods back into our diets. And he puts the role of fermented foods in the history of the world and its influence over cultures articulately by stating, "To ferment your food is to lodge a small but eloquent protest on behalf of the senses and the microbes, against the homogenization of flavors and food experiences now rolling like a great, undifferentiated lawn across the globe." So I encourage you to step outside your comfort zone and experiment with fermented foods for yourself. You might end up with a new passion, like me.